There we go. How about now? So that totally botched my opening joke, but that's okay. So I'm Ethan. I'm the founder of Perlstein Lab, and I want to tell you the story of how in the last 18 months or so, I went from washed up postdoc to founder of a lean biotech startup. You can learn more about us looking at our website, email us here, and follow us on Twitter. And you can live tweet this talk now. I don't know what you would hashtag it, but feel free to live tweet anything I'm saying. So we're a mission-driven biotech startup, and our mission is precision medicine for orphan disease patients. And by precision medicine, I mean personalized medicine, so specific to specific patients, even down to their specific mutations. And orphan diseases, these are often also called rare diseases, and that's the definition that's set by the Orphan Drug Act, which was passed about 30 years ago, and that threshold is 200,000 patients or fewer in the US. So some orphan or rare diseases you might have heard of include cystic fibrosis. Anybody heard of cystic fibrosis? Raise your hand. So a fair number. So actually that's good. I'll use that as a case study in a few slides to help clarify some of our points. So we're a mission-driven company, and what that means in action is that we are a benefit corporation, which is a new kind of corporation, and I can talk about that later in the Q&A. But our mission is obviously the unmet medical needs of these patient populations that have been traditionally neglected or historically neglected by pharma, but not just pharma, actually by academia too. So this is my story, and when you start off as a one-person startup, you basically have to sell yourself. So everything I'm going to say applies, obviously to me, but I do think there are some principles that do generalize, because I think most of us are all members of generation post-apocalypse, as I've been calling it. And this is my background. I was a postdoctoral fellow at Princeton, where I had my own small group, managed $1 million budget, and we were investigating the use of yeast as a model for psychopharmacology, and I'll explain a little bit more about that in a second. Before that, I was a PhD in Stuart Schreiber's lab at Harvard, where I was doing a lot of what they call academic drug discovery. And so that's my scientific background. And the milestones for Perlstein Lab, we're a young company, just really came on the map just at the beginning of this year. And we're located at QB3 at 953, which, as people in this room heard of QB3? So QB3 does a lot of things, but one of its responsibilities is to administer these incubators, and 953 is the latest iteration. I think there's 6 or 7, or 5 or 6 that came before it. And so I'll talk about how there are features of being in an incubator like this makes it possible to start off as a one-person startup and now be a 5-person startup in a span of several months. And we're affiliated with Janssen Labs, which is part of Johnson & Johnson. And that affiliation includes sort of marketing and also use of certain common equipment and other access to networking opportunities through Janssen Labs, which is really, really great and an awesome opportunity for us. And right now we're closing a $2 million seed round, and this is a snapshot from our AngelList page, so you could track our progress there. But you can only really look at the financials if you're an accredited investor. So this is the team. We had a new member join today, so she's not up on the website yet, but will be soon. So what we're talking about wouldn't be possible without the folks here and also the collaborations and the partnerships that are also depicted here. So one of the main things, in addition to the incubator that allows us to do what we're doing, is the ability to source experiments so that we don't need as much lab space, for example, or we don't need to buy as much equipment up front to do certain things. So Science Exchange, have people in this room heard of Science Exchange? There's a few. So I can talk more about that later in the question and answer, but they've played an integral role in helping us hit the ground running in terms of generating reagents and strains and other things for us that allow us essentially day one to get rolling. And there's a set of advisors, many of whom I met on Twitter. Twitter plays a big part in my story, and I think that's that's one of the general lessons I hope from this, is that if you don't have a Twitter account, get one now. If you don't have a website or a blog, make one now and start producing content. So the scientific basis for the platform, the drug screening platform that they're building, is based on many, many years that I've spent in academia thinking about how to use simple model organisms to illuminate complex processes. First, it was drugs, and I was interested in my postdoc in psychoactives. But the idea of using simple models to elucidate complex biology always was appealing to me, and it eventually occurred to me as I was leaving academia and figuring out what to do with myself that there would be a great marriage scientifically between simple model organisms, and the simplest of human diseases. These would be the orphan diseases that are genetic in basis, and so genetic that it's really one broken gene is responsible. So that would be like cystic fibrosis. So this concept, this evolutionary pharmacology concept, I've been thinking about and stewing on for a long time, and I think that's another key lesson of my story, is that a lot of ways I got to de-risk this idea in graduate school and as a postdoc, and that allowed me to, again, hit the ground running as soon as possible. And this is the problem that we're trying to address. There are 7,000 orphan diseases, but only 5% of them have FDA-approved therapies. So 95% of orphan diseases don't have any approved therapies, and that represents this unmet medical need that we're trying to address. And it also represents, in a market sense, a huge opportunity. And there's many reasons it's complex why the orphan drug approval rate is what it is. It's about 10 to 15 orphan drugs a year. If we keep going at that rate, it's going to take another 400 years before we have a drug for all the orphan diseases. So it's way too slow. And that lack of productivity is again, a result of a lot of factors. One of them is this, I think, overriding force called Arum's law. So maybe the people with pattern recognition skills here, because that's Arum's law flipped. And this is to describe the fact that it costs more and more money to discover and develop drugs. We're getting more and more inefficient, and it's taking longer. So now I think the averages are, $1 to $4 billion and 10 to 15 years to produce a drug, and the attrition rate is enormously high, sometimes over 90%. And many of these are candidates failing in the later stages of trials where you're actually trying to demonstrate efficacy. So it indicates that you probably shouldn't have been investing in this candidate because it failed. And it wasn't because it was toxic. It actually just didn't work. So your hypothesis about the disease and about the curative effect of this compound was probably wrong. But in spite of this, I think there's there's a ton of interest in the orphan disease space. Some people call it a bubble, but I think that represents a lot of forces, including the rise of patient advocacy, which I'll also talk about a little bit later. So our solution, from the scientific perspective, is a patient-centered approach where we try to appreciate the complexity of even these simplest diseases. And here's what I mean. So again, I said the rate of orphan disease is about 1 in 12, and this is a figure that applies across countries. There's going to be population genetic flukes where you have Finland or other populations where you have something weird going on, but in general, these frequencies seem to be consistent around the world. So this represents an orphan disease patient, and if this is a monogenic disease where there's a single broken gene that's responsible, like cystic fibrosis, turns out that there can be many, many mutations in the cystic fibrosis gene. It's called CFTR, which is a channel that sits at the surface of cells and conducts chloride ions. So it turns out there's many, many ways that you can break this one gene, up to 1,600 mutations documented. And that's sort of represented by this spectrum here. And our thinking is that you want to create drugs that are matched to specific mutations. We think that would be more efficient, but also allow a real possibility to address the entire population of orphan disease patients. Because it turns out it's, again, more complex than you think. It may be one broken gene, but many ways to break it, so you may need multiple silver bullets. Now, this approach actually works in the wild. Here's an example using cystic fibrosis as a case study. There's a molecule called Kalydeco, and these are the kinds of molecules that we'll be looking for in our drug screening hunt. This molecule is developed by Vertex with a lot of seed money from the Cystic Fibrosis Foundation. And it works in a really interesting way. It fixes the CFTR, the gene responsible for cystic fibrosis, that's broken in a particular way, having this mutation, G551D. And it turns out that this is a little schematic they lifted from Johns Hopkins that shows how these different classes of CFTR mutations probably are reflected in cell biology. So you have some group of mutations. In fact, the most common mutation is called delta 5-FO8, where you're missing this one phenylalanine at that one position, and that causes the protein not to fold correctly and never makes it to the surface, and it's sort of all gone, effectively just wiped out. But there are other mutations at different parts of the CFTR gene where the protein can make it to the surface, but one part of the protein's lesioned and it doesn't work. It turns out Kalydeco works by repairing that lesion effectively. And so this model works, and you can see that in the actual data, market data. So let's talk a little bit more about that market. In the US alone, we're talking 25 to 30 million orphan disease patients. Here's how they break down if you're thinking genetically. 80% of orphan diseases are genetic in origin, and if you think about that in how many genes are broken, we can start with the simplest case of one broken gene, and that actually represents the majority there. And about half of these single gene diseases, or these Mendelian diseases or monogenic diseases, pardon me for using different, it's my academic training that I have to use all these different words to describe the same thing. But there are about 1,500 of these single gene diseases that are diagnosed where we know what the causal gene is. That's like cystic fibrosis. Cystic fibrosis, CFTR. There's an equal number that all the genetics appears to indicate there's a single broken gene, but we just haven't identified what it is yet because the genome is this complex thing, and we're only now able to, with the drop in DNA sequencing cost, to plummet its depths, and we can now see, okay, this is actually the mutation that's responsible for this person's disease. And if you're talking about rare diseases, you may not have many samples to deal with. You want to have confirmation because it turns out our genomes are very noisy and we have lots of mutations. It's actually not trivial to correlate a particular mutation with phenotype, or in this case, a disease. So we think there's a huge opportunity to explore this monogenic space if you had a scalable drug discovery platform that could do that. And how would you do that? That's our solution, I think, which is using model organisms. That's not like, that idea is not itself new. People have been using model organisms, some like Thomas Hunt Morgan, back to the last century. But I think using them in the systematic way we want to use them, I think that's where we're really taking a new step. And I want to remind people that there is a lot of evolutionary conservation that's taken for granted. You think about budding yeast. These we're all eukaryotes, right? These are all eukaryotes. And so we have a last common ancestor with the single-celled yeast 1 billion years ago. And the residue of that is that almost a third of yeast, 25% to a third of yeast genes have a human homologue, a human analog, right? The corresponding gene. So they're related. And you can see how that ancestry just goes up the closer we come to us. And our platform is mammal-free. Our most complex organism is the zebrafish. And these happen to be all, again, well-studied model organisms that are used mostly in academic labs, not so much in pharma. They tend to focus almost exclusively on mouse models or rodent models or large animal models. And we're really taking advantage, again, of this convergence that's happening in the DNA space. On the one hand, it's getting cheaper and cheaper to do the sequencing. So you can go to a point where you have an undiagnosed patient, you can identify their mutation and very quickly start to do drug discovery. Because if you found the ancestral form of that disease gene in one of these model organisms, or all of them, you could start doing drug discovery on a very personalized disease model, because the other trend that's happening is that you can now edit DNA very specifically. So you can take those mutations that you record and then write them into the genomes of these simpler organisms and start doing drug discovery that way. Again, this has been done, actually. It just hasn't been scaled and hasn't really been applied to the orphan disease space. It's been applied in cancer, which is an example I'll tell you here. So Ross Kagan is a researcher who has used, he's now at Mount Sinai, a professor at Mount Sinai, and he has used flies as a model for drug discovery in cancer. How did he do that? Well, first of all, let me skip to the punchline. This is the drug that resulted from a screen that started in flies. And this is a thyroid cancer drug. Flies don't have thyroid glands. But it turned out that Ross Kagan had this realization, well, let's just take the gene that is causing this cancer, this thyroid cancer, it's a single gene, and place it in the fly eye. This is the normal fly eye, and this is a fly eye that's expressing that thyroid cancer causing oncogene or gene. And all he did was look for small molecules, compounds, these things, what we call drugs, but technically, small molecules, that can rescue this and restore it back to normal. Restore it back to normal at this very fine ultrastructural scale. In other words, you're zooming really fine structure, and you're seeing that this one molecule is taking this incredibly complex disease state and restoring it essentially back to normal. That's pretty cool. These are the kinds of molecules we want to find, but targeted in the orphan disease space. And this is the platform summarized in one slide, and we call it lead gen for orphan drug developers. You can start with, again, whether you know the mutation because it's a known Mendelian disease and you have a database sometimes of mutations, or it's hot off the genomic presses and you have a causal gene that you've identified and you want to now kickstart drug discovery. However you start with the input, you then use CRISPR to dial in those specific mutations at the ancestral forms of the disease gene in as many of these organisms where that ancestral form is present, and then you do these rapid, scalable, growth-based screens using technology that has gotten better and better over the last decade or so, but again, it's been around it's not new per se. And then we'll do lead optimization and validation studies in patient-derived cells so we can kind of close the loop here, right? So starting with these mutations in a sequence sense, putting them into these simple models, and then we want to confirm that in essentially the same mutation, but in the context of what is the patient's most similar model. And we're able nowadays to take patient cells, even sort of samples from blood or from just skin biopsies, and convert them into a number of different differentiated cells or different cell types. Not all of them, but just specific kinds of neurons, you can do this. And that, in a lot of cases, that might be the tissue that's really manifesting the disease most. And that's where you want to see if your drug candidate, is really working. And by the way, we think we can do this really quickly because we can take advantage of the fact that these are rapid, many generation, fast generation time, rapidly growing organisms. And we can still do whole organism-based screens, which I think is a really powerful phenotype that we can really leverage. We're focusing first on the lysosomal storage diseases. There are about 49 of them, and 47 of them, there are ancestral versions of those genes across these models that we're using. There's only two of them, two lysosomal storage diseases where the gene only goes back to mammals. So we can't do anything about those right now using these organisms. That's the caveat. And this is how the diseases are distributed. We're starting on this end here, which are the diseases where the gene is really, really conserved, sometimes down to the level of the actual amino acids that are being mutated. So the amino acids that are changing, that are causing disease, are actually some of the most conserved amino acids when you look at these ancestral forms and compare them. So that's also, I think, pretty revealing and validating of this evolutionary perspective. But we can start here and then sweep across this space. These are all the diseases that you can only study in fish. Those are going to be a little logistically hard to do. We can set up at our QB3 type environment. We can set up fly, worm, and yeast work pretty straightforwardly, or at least that's our hope. But I think there's plenty of labs that have combined them in an academic setting, so we think that we can do the same thing. So hopefully I've roused some of you to join me in an orphan disease moonshot, and I'd be happy to take any questions. But first, I want to show you one way that I think we're going to be different than maybe a lot of other biotech startups, and that's through our approach of online engagement and online outreach. And we want to engage with patients, we want to engage with the public, we want to engage with advocates, and we want to do it in ways that are accessible, not condescending and fun. And I think one example of that is potentially to use rap to explain science. So I want to show you a video that was made by a rapper named Baba Brinkman, who took the idea of evolutionary pharmacology and explained it much better than I can in spoken word. So I'd like to share that with you, and then happy to take any questions.
I'll make it simple, so try to follow me. This is evolutionary pharmacology. The human body is a branch on a tree that goes back to the ancient roots of biology. They say a monkey can't change his tail. Well, we're descended from monkeys and other things as well. Some things change, other things stay stale. A couple billion years back, we were single-celled. We mutated into fish with scales and then went amphibian and grew legs as well. DNA replication just to gain some scales until the time came for brains to swell. And now compared to the past, our minds are gigantic, but you can't expand it without the cell's mechanics. From vesicle formation, we made it to synapses. Pharmacology's all about the same dynamics. We take drugs, stimulants and sedatives, used as directed with confusing effectiveness. But the Perlstein lab has a new perspective, treating yeast cells as a model of ancestral kin. Yeast, you can make beer in a bottle with it, but it's also kind of a living fossil. Just give it a pharmacological dose and watch it respond and become an Evo Pharma apostle. And I don't mean to profit on a soapbox. Mental illness is one of humanity's main roadblocks, but one lab is breaking new ground by feeding yeast antidepressants like Zoloft. So what does Zoloft do to human beings? According to the box, it blocks the proteins that uptake serotonin, and a brain's a fiend, so more serotonin means more well-being. Well, that's problematic. Depression has to be more than a chemical imbalance of serotonin. Otherwise, antidepressants would work instantly instead of taking on average several weeks or months before they kick in. The effects of the drug should be instant, cause minutes later the serotonin level is different. By the way, don't give Zoloft to infants. Okay, hypothesis, test 1. They got it published in PLO S1. Zoloft accumulates in yeast cells, which is odd cause they don't have serotonin expression. It even causes their shape to change. It twists up their vesicle-forming membranes. So might that explain the strange time lag effects of antidepressants in our brains? Maybe it accumulates as an obstacle, or maybe it causes new neurons to grow. That happens in rats, so it's possible. But either way, I think we all want to know. That's evolutionary pharmacology, comparing drug reactions between us and yeast and rodents and new targets for the drugs to treat, making progress to stop disease. See, our distant cousins can't teach us how to focus. We gotta capture this collaborative moment. You ain't even gotta cross the Atlantic Ocean to find out, cause now the whole lab is open. Perlsteinlab.com. You can log in, check the results, and get the problem solving. Use your noggin and get involved, cause we blogging. That's how knowledge keeps on evolving. Yeah, this pharmacology, it's evolutionary pharmacology. That's evolutionary pharmacology, it's evolutionary pharmacology.
And I should say, if anyone has any fly questions, you should ask Tom, who's back there over there. Say hi. He's part of the team as of tomorrow officially. So yeah, happy to answer any questions. Thanks for your attention. Yeah, so it started with doing being a consultant, so I could do part-time work and pay some bills. That helped. So I was fortunate enough to team up with Mycorrhiza, now experiment.com. They're like the Kickstarter for science. If you don't know them, you should check them out. Back some really cool science. So yeah, I started out there last year and did 6 months with them, and that also allowed me to stew more on the business side of things, because the science I thought I had a good control over, but stewing on the business side and talking to confidants and consultants, that took some process, and then the process of getting feedback on a business plan and on a slide deck, that took several months. But that kind of started after I stopped working as a consultant and just realized, okay, I just got to go, just go for it. And that was last fall. And then, getting an investor through a tweet, which was totally serendipitous, but that's how it kind of ended up happening. That process of fundraising really hit high gear starting in the beginning of this year. And so then the team really came together in April, and here we are in May. Yeah, so those options would be like the SBIR and STTR, and for us, I think they're just too slow. The grant cycle is just too slow, too cumbersome, and the pay lines are too low, and I don't see any radical change in the budgets happening for the federal government, research agencies until 2016. I mean, realistically, I think so. Yeah, we're pretty happy with the way we're getting investment. And yeah, I think if grants were faster, non-dilutive capital is great. But yeah, I definitely wouldn't discourage people from those opportunities. But that's just not the path we took. Yeah, I mean, we envision a hybrid business model where we're developing some candidates internally, in which case I guess the customer would then be the actual patients or advocates, something like that. But we can also imagine partnering with orphan drug developers, and there are a number of them that are already in this space, and potentially be lead gen for them. So in that case, it would be a B2B model. But again, it's kind of hybrid, where we want to be able to internally develop drug candidates as well. But we think we're going to generate a lot of them by the very nature of using model organisms and the fact that they're fast growing. In terms of funding, and that's up to the way money flows into universities. And right now, that raises the big indirect cost question. For us, the overhead is the overhead. We see what it is. This is the rent. But in the case of academic labs, you've got that layer in between. So academic labs generally don't just receive hundreds of thousands of dollars without the university being like, oh, really? So you'd have to solve that problem. And you'd have to, I guess, convince some investor on a longer time horizon, and that is a trend that they call it venture philanthropy. I think basically, people who would consider a longer time horizon than they've gotten spoiled with maybe some tech exits. But that's also, I guess, another a potential avenue for academic funding. But I think it's still, you have to sort out a lot of administrative issues, and that tends to be very local from institution to institution, with some institutions being more progressive and some being more hidebound. So it all sort of depends. No, so the crowdfunding part of the story was the charitable crowdfunding we did on Rocket Hub in fall of 2012, where we raised 25K for a psychopharmacology project that's happening in a collaborator's lab at Columbia. The idea for Perlstein Lab was to go on AngelList and try to rouse support and get investors or investment that way. But just the way things shook out with that tweet and how that led to a chain of events where ended up having a lead investor, that I sort of focused on the hand I was dealt. And so the idea of trying to crowdfund it all while appealing, I think there are places for crowdfunding. There are certainly startups like Ubiome, which have raised, I think, $1 million using AngelList. I say using AngelList. The transaction doesn't happen on AngelList like Kickstarter, but that's an example of equity crowdfunding that I think is one possible source of funding, and something that we haven't, we're still open to that. The vision, I think the vision is to stay independent and eventually IPO, not an M&A play. We're raising 2 mil on a seed round, right? But the idea is that just like a tech startup would, you hit milestones relatively quickly where you can then justify a higher valuation. Normally, biotechs have to take a lot of money up front. The average Series A is $15 million. They take all that money, spend it on all the capital-intensive stuff they do, and then either their bet paid off or it didn't, and they need 3 to 5 years to figure that out. I think we can do things on a much faster time scale. Competition in what sense? You mean people would self-edit? Sure. Yeah, that's the bet. That's the $43 million bet that Editas is trying to pull out, which is the genome editing company. That I think that's their goal. I don't see that happening anytime soon. I think small molecule solutions can originate, on a 6 to 12 month time scale. They can be developed, on a 3 to 5 year time scale. If you're talking about repurposing, that can, you can initiate efficacy trials, relatively quickly. So I think small molecules is another reason why we can do things more quickly, because I think that is the most de-risked of all these various therapeutic approaches. And God bless gene therapy. I hope it works, but I just don't see it happening in the next 3 to 5 years on a patient-by-patient basis, on a disease-by basis across globally, with the potential to be truly global solution. Yeah, so in some sense, we're like any other drug discovery company. It's really about getting IP around specific chemical matter, right? Because that's the drug you're going to patent. That's the Lipitor, right? In the current model, the current blockbuster model. So in that sense, I guess it's kind of that's not where the innovation necessarily is. I think it's in other aspects, which include, I think, the way we envision partnering with patient advocacy groups. Because I think the real cost of bringing a drug to market is in the development side and the clinical trials side. We have to make those faster, smarter, cheaper. And so we're trying to do this drug discovery approach, which is the preclinical side. But we need to, for our fast approach to work, it's got to be partnered with a fast, smart clinical trial arm. And I think the way that's going to happen is by bringing the patients in from the very beginning and watching how their mutation gets put into a model, which then gets turned into a drug candidate and so on. So they understand when they're going to FDA, this is what this is at stake. And I think that would enable this process to happen more expeditiously. And FDA is already doing things, already enacting reforms to, encourage and incentivize orphan drug development. So we've got that tailwind as well. But I think the aspect of using model organisms specifically, there's a company called Exelixis that was founded, 20 years ago or so, that was based on using worms and flies to do drug discovery. But they picked cancer as a target. I think that was just too massive a problem. especially back then. And orphan diseases were not really on anybody's radar screen. But now is their moment, and I think therefore it's also time to bring back the model organism approach, especially because we've gone so down far this mouse hole where everything is essentially mouse model, mouse model, mouse model. No, 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 no. This is about Yeah, I mean that we're trying to get to a point where we can develop some drug candidates that we can either be promising enough that would enable IND studies. The apex. Yeah. So how many? I mean, I think it depends really on how the mutations sort out. I mean, is it going to turn? So we're initially starting with 10 mutations that represent, what we think is a large portion of the patient population. Does each one of those mutations require its own compound, its own prodrug? Don't know. Maybe the 10 actually they all converge on one. I doubt it. There's a lot of evidence that suggests that's not going to happen. Probably going to resolve into 2 to 4 classes, in which case, here's your answer. But it would depend. It also depends on the throughput. How many diseases can we do? That would be per disease. The whole point here is to do these kinds of screens in parallel. $100%. 100% backed by many a Cell paper, if you don't believe me, because it's all just really applying classical genetic ideas, but really to the hilt, and for the orphan space, with an eye toward a scalable solution, not just another, you can't keep doing 10 to 15 orphan drugs a year. It's going to take another 400 years. We need to do this in our lifetime, my lifetime. That means we need to be at 10x that. I think if you're starting to parse these diseases by mutations, and therefore by patient classes, that's one way you can start to turn up that dial. And then, of course, being able to do multiple diseases in parallel, that's where you also start to get, or maybe we're going to go to an era of 100 to 150 orphan drug approvals a year. That's cooking with gas. No, I expired. My 5 years were up. Our competition is the mystery of these puzzles. I mean, the more the merrier. But the reality is that, yeah, I mean, I think it's still, hopefully when we validate this, that would inspire others to come on board. It's a 7,000 disease space. We can't possibly, unless we turn into factories of, unless we turned ourselves into fish and fly, we can't possibly do this alone. But I think in fact, I find that startup land is actually a relief from the hyper competition of academia. People are way more likely to actually be collaborative and volunteer their time. And we all know that their time is valuable because everyone has a consultant rate. And people will voluntarily give you so that, I've encountered that way more than academia, where oftentimes it would be difficult to get a professor to respond to an email. 10% of the time, it bounced because probably that person left. So I find startup land, in that sense, way more collaborative, open. Doesn't mean it's more egalitarian, doesn't mean it's more equal, but I do find that there's a difference in that competitive, or in that sense of competition. So anyone know what a B Corp is? Okay, we've got some lawyers in the house, maybe. So maybe I'm going to screw this up. But no, I think it's a law that started in 2010 in Maryland, and it's really, it's kind of cosmetic in a lot of ways. I mean, practically speaking, it's just an extra little blurb in your articles of incorporation talking about balancing shareholder profitability with the social mission. Some people refer to it as the triple bottom line. So it's now, I think, in at least 20 to 25 states, set to be 30 by the end of the year, and that includes California. So Patagonia is a B Corp. In fact, on the first day B Corps were allowed, Patagonia was, I think, one of the first California B Corps. But there are only 1,000 B Corps in aggregate across the country, so it's still very new. but I think it offers the opportunity to put your money where your mouth is in some sense. If you're going to be doing patient-centered approaches, we're getting to a very heated debate nowadays about the cost of these drugs, and $300,000. That Kalydeco is $300,000 and change per patient per year. And we're on a kind of a runaway train to $1 million per patient per year. But back in the day, 1986, 1987, AZT was the most expensive drug at $38,000 a year. I mean, that's just, you think the price of college has gone up. Well, it's also the price of these drugs, orphan drugs have gone up. So we've got to reverse that trend as well. I think that just comes from more competition, but anyway, that's another discussion. But yeah, B Corps are this new trend in corporate law. There's also things called L3Cs. I learned all about this from my brother Henry. He's the lawyer among the brothers, and also our web developer. So we can say that Perlstein Lab is a family business. So I mean, he could speak more to this, but there are these L3Cs, which are kind of LLCs merged with 501c3s and got this hybrid character of both. So there's all kinds of cool innovations happening in corporate law, and I was captivated by this B Corp message. And so, yeah, we're a public benefit corporation. I mean, we're only going to advance so far to basically the pre-IND stage. So we haven't spec'd it out that far because part of the uncertainty in the costs are how much can you really drive down the cost of these trials. If you can avoid having to fly everyone to some central location. You're already talking about, in these orphan diseases, the geographically dispersed populations. So they're kind of crying for remote sensing and quantified self kind of technology. That could dramatically bring down the cost of these trials. So it's hard to spec out what it's going to take, I think, post-IND. But I think getting up to pre-IND and then getting from pre-IND that development to IND, we need to raise more capital after this. The $2 million is not going to go all the way to IND, but it's the investment in the IND, or the down payment of the IND. I don't know if it's going to necessarily. I mean, it's going to end up, like with cystic fibrosis, that one allele, that's still 4 to 5% of the cystic fibrosis patients. There's 150,000 of them worldwide. So you're not talking about insignificant numbers where it's not down to the point of literally one. But I think, a disease like progeria, I don't know if people are familiar with progeria, but this, rapid aging and degeneration, but otherwise sort of cognitive function totally fine. That's 1 to 200 estimated aren't in the world. So, no one has a business model to do that. But then maybe that's where you have nonprofit biotech. So I don't know. But right now we're focused on lysosomal storage diseases, but we do have an eye toward expanding. But yeah, the idea is initially this proof of concept is going to take us to a pre-IND enabling set of candidates. Not sure we're going to be competing with them right away. I mean, I was at Mayo Clinic not too long ago, and that is an example of a place where you would potentially conduct clinical trial, or at least you have the where the clinicians are full time, and so we're exploring opportunities there. Nothing has happened yet, but so I think I don't necessarily see it from the point of view of, I guess, competition. I mean, for us, it's really about how do we get patient-derived cells that match the genotype that we're examining, and either those exist in biobanks that have probably been set up and maintained by the patient advocacy groups or some designee, or we have to get these samples de novo, and they're actually mobile phlebotomists who will go around and collect blood at your home, and so there are ways you can get the sample. So I'm not sure I see it in the lens of competition, but maybe I didn't quite understand your question. Yeah, I mean, initially we're starting with lysosomal storage diseases in part because those are some of the most well organized of the communities. And so a lot is sort of known about where everybody is. They may not have all been biobanked, for example, but the groundwork has been laid in some sense. And my experience interacting with patient advocacy groups, they really want more scientists to be interested in their disease. There couldn't be too few scientists interested in their child's disease. That's I think the sentiment. So, and I think they're kind of starved for it. My experience going to certain conferences where it was basically just the NIH policy folks and the NIH scientists and the patient groups, and just no like basic researchers were there. They're just a little bit horrified by that, but it shows you that it's not just pharma that is ignoring orphan diseases. But grants tend to have to be justified through some common disease impact, and so you end up neglecting somebody. Yeah, I mean, I think obviously, as I said, the value proposition here are these chemical structures that you're linking with some therapeutic potential. Yeah, we're not going to be tweeting those or Instagramming those, but I mean, for us, we're a platform play instead of an asset play. So with the assets, it's usually that's protected by some intellectual property. For us, the platform is really doing genetic screens, and that's just not really patentable. In fact, actually, it was patented in 1990, so it's now in public domain. So it's basically one of these cases where, in some ways, it's just this idea that's under everybody's nose, but nobody's really doing anything about it. Even though every aspect of it is really individually makes sense, and you can show case studies where it works, and these case studies don't suggest that these are one-off things. So, but yeah, I mean, we're going to have to, as being active on Twitter as I have been, it's been in a lot of ways in service of open science, and a lot of other people I think feel that way. That at least I'm connected to with Twitter. So, I think there's certain principles that I want to abide by in that sense. Only publishing in open access journals. They would never publish in a closed access journal. I can make that promise now. I signed the Mendeley, or not the Elsevier ban or boycott. So I can come out on the record saying that. But yeah, there's going to be some cases where it's proprietary or trade secret, and yeah, we're not going to necessarily reveal that. But I think in time, yeah, I do think with some embargo, some data will be able to release in the service of furthering research for this community. And I think that would be in response to feedback and input from the patient groups, because again, I want them to kind of be vetting us, sort of from the beginning. Yeah, so I started on Twitter in January of 2011, and my impression of Twitter for many years was like, oh, that's where Justin Bieber tweets about stuff, and I just thought it was ridiculous. And then someone kind of alerted me to the fact that scientists were on there talking about science. I thought, well, that's odd. So then I joined, and it took a long time to figure out how to use Twitter, and then it didn't take very long to get addicted to Twitter. But I cut back on my Facebook consumption. So it's still balanced. But yeah, I mean, Twitter has been a place for me to just find people and network. And part of it started when I was on the tail end of my postdoc and kind of realized, oh, I was starting to go stale, and there had been some internal palace intrigue at where I was, and so it wasn't and I was commuting, and so in a lot of ways, Twitter was kind of like a support group, because I was finding it basically, I think the beginnings of what is now post-apocalypse, at least rumblings of it, but also the open science movement and others who were really starting to organize on Twitter when no one else was really paying attention way back then, probably even before I joined. But yeah, but then it took many years to really get to where I am now, in terms of being able to, I guess, monetize Twitter. But I don't think about it that way, because I just think I'm using Twitter the way a lot of other scientists are using Twitter, which is, and a lot of other people, in fact, which is just kind of either chime in on interesting conversations and be a fly on the wall. Maybe don't chime in sometimes, but still get to see these amazing kind of little conversations happening. And if you use it that way, and I think you get standing in the community, then your following, yeah, is powerful. But I didn't anticipate finding a lead investor through a tweet. I And I think if you tried to do that intentionally, it probably would fail. But it happened. But I think I've also sent random emails to people years ago who end up becoming amazing mentors. So some things just sort of start this way, and sometimes now it's Twitter. Back then it was email. But these random connections, ability to and in a way that seems pretty meritocratic, I think, where a lot of parts of the internet are very noisy and rude, and Twitter has its fair share of that. But I think if you devote if you commit to kind of the fact that it's a community, but you also meet the people there in real life. I think if you just do it virtually, to me, it doesn't make sense. Then you justify also the time. But I think Twitter can be an amazing platform for networking and reaching out to people of like mind. Sometimes, I mean, you can kind of tell. It's just like if you're in real life and you're, at a mixer and of a conference, and people are broken up into little cliques and they're having their conversation. You can kind of walk through and listen, and sometimes you don't jump in. Sometimes you can tell that sometimes it's an open, private conversation, and you have to learn the etiquette of that. But most of the nice thing about Twitter is that people are themselves, for better or for worse. They are themselves on Twitter. But yeah, it takes some time to get comfortable there and realize what it's useful for, how it's useful to you. And so again, I guess I'm a power user. Probably certainly am. So maybe I have a skewed perspective, but I have interacted with all kinds of people on Twitter. People who are like also diehard Twitter people, also people who just only chime in like when they have the exact right thing to say, and it's always a zinger. Like it's just another community. But for me, you have to connect it to real life. You have to meet these people. Although there are certainly people out that are like on the other side of the world I haven't met yet, but I feel like they're my friends, just because we bonded over open science or whatever it is. Definitely more intimate than LinkedIn, in a good way, I think. Yeah, I mean that was actually quite key because I joined Experiment when it was at its when it was basically two people with like remote designers. So I got to, and then I watched them get funded. I watched them do the Y Combinator thing and watch them start to take off. Watch them think about week over week growth and all these other things. And so that was actually invaluable experience. And then I was also active on Twitter, networking with people, and a lot of the listening in was like trying to listen in on expert conversations where initially you're like half of it is jargon and you just almost follow it, but if you knew what those three letters meant, and then after a while you know what that means, and then you feel like, oh, I can now listen to these people's conversations, and it's like really high caliber stuff, and they don't even know it, and then you can sometimes engage or not. But yeah, I got definitely a lot of advice and concrete feedback through those interactions, but also, I had a set of peer reviewers who looked over my business plan and slide deck. A lot of these people volunteered, their expensive time to do this for me, based on a recommendation or a request from a friend, or based on the fact that we had been Twitter buds, and that was invaluable. Um, no, no. Yeah, no. I really wanted it, and I wanted it for a long, long time. I mean, I would have killed for a first author Nature paper for many years. I'm not ashamed to say it. Maybe some of you relate. No, I can't regret. I don't regret a single. I don't. I couldn't be happier now. But at the time, I didn't know about. I didn't really know about startup life. I mean, so. But I think, yeah, this is my version of this is my story. But I do hear a lot of whenever I tweet something about post-apocalypse, like people retweet it and people favorite it, so it's resonating, not just in my head. But yeah, I think maybe someday I'll be an adjunct when it's actually the right use of the adjunct, not as the way to get into academia, but like when you're actually you have a day job, but this is like this other thing you do. But yeah, I couldn't be happier now, and forward.
are really interested in. And thank you so much for coming. Joy, and please welcome one more time Ethan for this.
Sorry, I realized I took the whole time, but I guess.
No.